All right, everyone, we're going to go ahead and get started. Thank you so much for being here. My name is Cinnamon Moffett. I am the Research Program Manager at Oregon State University's Hatfield Marine Science Center, and I welcome you all to this afternoon seminar. For some possible new faces in the room, this is a hybrid event, so we have folks online and folks in the room. For folks that are online, we have your cameras, mics, and screen shares disabled, but we ask that you put any questions you might have into the chat box, either because you're having technical issues or you have questions for our speaker, and our volunteer Roseanne will help out and make sure that those questions get resolved or heard by today's speaker. For folks in the room, if you have any questions, just raise your hand and I'll bring you a mic, and that way folks online can hear your questions as well. I also just remind you, if you are a student, to make sure you log in if you're in the room to the sign-in sheet outside the door. That is how we make sure that we know that you are here. So just a reminder there. So I wanted to do a quick reminder that next week is a holiday. Yay! So no seminar, but the following week, on November 30th, we have Carl Hendrickson, who is here from the Department of Land Conservation and Development. He's going to be talking about a project working with bottom-up community engagement to prepare for climate change impacts like sea level rise. So he's a really interesting speaker. I hope you all come back after the holidays. But the reason you're all here today is for our speaker. And this particular speaker was invited by a familiar face, Emily, who many of you know. So I'm going to hand this mic off to Emily, and who's going to introduce today's speaker.
Hi, everyone. For those that don't know me, my name is Emily Schlesinger. I'm over at the Alaska Fisheries Science Center, and I'm very excited to introduce our speaker today, Dr. Nick Battista, who's both a cherished colleague and partner in life of mine. So if you ask Nick who his favorite scientists are, he would say they're the Ghostbusters. And so it would be no surprise that Nick double majored in applied math and physics at Rochester Institute of Technology, where he stayed for a master's, then transitioned to UNC Chapel Hill to get a PhD in mathematics, working on the fluid dynamics of heart development. And then he went off to the College of New Jersey and as an assistant professor, and has recently leveled up as an associate professor, where he took tools that he learned in his PhD as well as some software that he developed, but started asking completely new fluid dynamics questions, focusing on swimming behavior of marine invertebrates. So overall, Nick is really excited by the biological world and explores questions in it through the lens of mathematics, which I think to a lot of us biologists can present some really unique and new novel ways to ask questions in ecology and evolution, as well as develop different types of software and applications that he uses all the time, and that some of us are starting to think about using now. And just a quick plug, some of us are going to go to happy hour at Beer One after this, so if you want to berate him with more questions over a beer, you're more than welcome to. All right.
Thanks for that very nice introduction. I think that's probably the nicest introduction I'll ever get for any talk, so we're only going down from here. But yeah, my name's Nick Battista. I'm currently in the Math and Stats department at the College of New Jersey. We're a small, primarily undergrad institution that's nestled about halfway between Philadelphia and New York City, if you're wondering, over in the Northeast. I'm excited to share with you a little bit of what my undergraduate lab and I have been thinking about, oops, spoilers, for the past couple of years involving animal locomotion, specifically looking at how the biomechanics of swimming for a variety of different invertebrates. We use a blend of computational modeling, computational fluid dynamics, as well as some machine learning approaches. So without further ado, here's really a subset of my team from the past couple of years that are really responsible for a lot of the work I get to share with you. So again, these is a part of my undergraduate research team. In my position, I get to work with a number of undergrads from a variety of majors, from, of course, math and stats right in my home department, but other sciences, biology, physics, computer science, as well as various flavors of engineering, and even some majors like communications. So a nice thing about my lab that I really enjoy is we bring together a lot of different perspectives where students can bring their own kind of personal flair to our group using techniques that they're learning in their own disciplines. And hopefully we can start looking at some integrative approaches to some of the problems that we think about. And most of the things that we think about all kind of fall under the heading of what's called a fluid structure interaction. And this begins the nerdy portion of the talk. Now, rather than sort of give you the real nitty-gritty mathy type definitions, I'd rather just give you a visualization to help you guide you through this process. But you could have things like these hearts, these ellipses that resemble jellyfish in a moment, or these letters, and you can think of them all sitting or being immersed in a fluid environment. And for our purposes today, you can think of the fluid, whenever I say that word, just as something like water. So what our group tries to do is we look at how the interactions, the movements of these objects, whether they bend, maybe they flex, or they're trying to swim. We look at how those interactions with the fluid that's underlying in the system, how those interactions result in some type of swimming behavior, whether that's good, whether that's bad, and we try to address problems in biology through this type of lens. And as you can see, as the simulation progresses, so this is a very contrived simulation, is you can see that there's a lot of highly dynamic information that comes out of these types of interactions. So these different colors in this case represent fluid vorticity, where the orange or the darker black colors represent basically opposite spinning vortices, a counterclockwise, clockwise vortice. And you can see that these vortices form, they interact. There's a lot of information that we can start gleaning from these types of simulations. So in today's talk, I want to do a couple things. The first is just kind of give you an overview of the types of problems that we tend to think about in our group. What are kind of the biological questions that we're kind of underpinning everything we do? And then I kind of want to give you a walkthrough of the actual math modeling process that we use in our group to kind of give you, maybe share an applied math perspective of how I approach these types of things with my students. And hopefully there's times that we can actually dig into some of the results and really explore some of the data that we curate. All right. So let's dive right in. So one thing that's real fun about the type of work we get to do is we look at a variety of different animal systems, and the ocean's just chock full of a highly diverse array of animals, both in morphology, size, and shape, as well as just different scales as well. So, and we always think about how the animals in these types of harsh environments kind of solve the problem of just moving around and doing the things they do kind of on a daily basis in these types of water-based environments. So we've been coming through this with the lens of how these animals swim for a little bit now. So you have animals like this jellyfish on the left that's using some variation of a jet propulsion or maybe jet rowing, kind of jet paddling technique. There's a whole bunch of animals that use, like the shrimp in the middle, some form of rhythmic paddling, where each paddle is sort of doing slightly off the leg or the limb that's next to it. So this is called metachronal paddling, where each limb is sort of a little time off from the limb in front of it. And a lot of other animals use this type of mode as well, from other types of crustaceans to even things like ctenophores use this type of paddling. But then you might be more familiar with this type of undulation that a lot of animals exhibit in the ocean. So this is a giant larvation here, but I'll just point out there's a body wave that kind of travels along the body of this animal. And this is the body wave's direction is going head to tail in this case. Some other animals that display this are basically all of your, oops, are we freezing? All of your kind of fish, from things like eels all the way to sharks. Even things like crocodiles will display this in their tail, or some marine mammals sort of in their caudal fin as well. So lots of animals use some variation of this bodily undulation to some extent. Okay. But some of the other animals that we've thought about in our group have kind of developed really complex, intricate ways to kind of solve this problem of actually moving around in the water. So from things like the siphonophore on the left-hand side, where it looks basically like this organism's just 20 jellyfish kind of all glued together or so. But each of these jellyfish-like bell structures is an individual organism. So this is a colonial organism. So there's lots of interesting questions you can begin to ask in terms of its collective behaviors. How do we coordinate kind of the pulsations of each of these individual nectophores to achieve different tasks, whether that's just a forward swimming, whether that's maneuvering, or some type of foraging ability. But the animal I want to focus on a lot of today's talk is this polychaete. This is Tomopteris on the right-hand side. So this will be the animal that I kind of walk through the math modeling process in a little bit. But for right now, I just want to point out two interesting facets of this worm. You can see that it's using a very nice rhythmic paddling with its parapodia on both sides of its body. So it's doing a nice metachronal paddling process, as well as its body's also doing a nice undulatory or wiggling motion. So this animal is doing two modes of locomotion together at the same time. So there's some biomechanics questions that we'll dig into about how that may be useful a little later. But in general, the types of problems that we tend to think about in our group all fall into this category of we really think about how variations on an animal's morphology, as well as its variations on the way it tries to move or its kinematics, how those types of variations can affect its ability to do different tasks underwater. So we're basically asking how morphology and kinematics affect its performance. And when we start thinking about performance in a biomechanics framework, this is a pretty loaded term. It can mean a whole lot of different things. You could just maybe be thinking about performance in terms of a steady forward swimming speed, maybe how efficiently it's swimming around, maybe its ability to maneuver and turn. And the idea with these types of performance metrics is you can also try to cast these into a greater ecological context. In terms of if we're thinking about its ability for quick accelerations, that can kind of give you a glimpse into maybe the animal's ability to escape from predators, or maybe to actively and appropriately hunt for its prey. And you can also think about animals creating things like feeding currents to also draw prey near it. So just thinking about performance in terms of various metrics can also give you a glimpse into how these animals are actually behaving and what they're doing. And you can also cast these types of problems in the lens of an evolutionary lens as well, when we start thinking about how variations on size and shape and that type of thing can kind of cast along kind of evolutionary lineages. If you can start playing this game of maybe just moving a limb slightly over, developing more limbs, and that type of thing as well. So our group has done these types of variational studies for a variety of animals, from different types of species of jellyfish to different invertebrate undulators to even different types of paddlers like this copepod. So my undergraduate students and I try to think about what are some of the salient or important features of all these swimming systems in both their morphology as well as their kinematics. And my undergraduates will create computational fluid dynamics models that try to mimic these types of behaviors that we think are important for the system. So from there, we can start playing this game of once we have some of these models up and running, how do variations affect its ability to perform different tasks? Right? And we can also think about this in the context of maybe things that are closer to home here in terms of fish studies, but we can also start thinking in perhaps a more interesting note of the actual underlying physics of some of these collective behaviors where you start thinking about fish schooling or fish shoaling. What are the actual benefits for different configurations, maybe at different scales, different morphologies? How are fish actually trying to behave, and what benefits do they actually get from that? Now, before I get too jazzed up here, because I love math modeling, I love making these models, asking kind of biomechanical questions, we also need to validate these models, because we don't just want to be making stuff up in a mathematical sense, seeing, hey, that's cool, but it may not mimic anything we actually observe in biology. So one way in which you do this is you compare your actual fluid dynamics simulations against data that's out there from actual organismal experiments. So just to kind of give you a quick glimpse into this world, on the left-hand side is a snapshot from a paper from Brad Gemmel's group in 2015. This is a juvenile lamprey. The background colors is what's happening in the fluid pressure as it's swimming. And basically what I happened to do is just run a simulation that had the same general morphology and same kinematics as this lamprey. And just as you can I tried to match the same colors in the color map, you can see the same fluid dynamic signatures are there in the pressure field in this case. So we try to do in terms of validation is try to make sure we're observing the same underlying physics of the system. And then once we have sort of the signature model pinned down, we can start asking some of our variational questions. So on that note, I want to actually just walk you through sort of a day in our life of how we actually approach these types of problems. So the first thing that I want to discuss in terms of these types of applications is we might see a really gnarly animal and want to start asking some biomechanical questions. So how do we actually start by having a targeted biomechanical hypothesis, as well as starting to put together a model to describe these systems? So the model system we'll use or organism we'll use is Tomopteris today. So I'll just point out a couple more unique features about this polychaete. It is using metachronal paddling Each side has rows of parapodia. And as you can see, each side parapodia are not doing synchronized with what the other side's doing. They're kind of doing a nice offbeat of each other, as opposed to something like the breaststroke. Similarly, they're also doing this all in a 2D plane. So their parapodia are outside, and they're all doing this in a 2D motion. So turning side like this, do more of this dancing. There is some 3D motion with their rami at the end of a parapodia, kind of doing a Spock-like Star Trek thing. We're doing a power stroke. They're kind of opening them up to push more fluid, and then they're retracting them on the recovery stroke. But most of the actual motion is very much 2D in a plane. They're also doing this bodily undulation, like I said before, this wiggling. However, if you stare at this video long enough and stare at fish videos, there's something really interesting about this motion of the body wave. It's actually going the opposite direction of what we'd expect for forward swimming. So what I mean by that is if we put something like this Tomopteris and an eel side by side, their body wave motions are going in the opposite direction. And eels is going head to tail, whereas Tomopteris is going tail to head. So just from our simulations, if we want to chop off all the parapodia and still have that same body wave dynamic, this thing will just swim backwards. So an interesting biomechanics question is basically, well, why are they doing both of these modes of swimming if one mode seems to be pulling it downwards, whereas another mode is trying to push it forwards? So why is this animal doing both of those things? And we can generate some hypotheses on why it's doing that. But that's sort of the targeted question we want to address. How can we explore these dynamics of two modes synergizing for some type of swimming? So when we approach that type of question, we want to start creating some type of computational geometry that mimics the movements. And just from the math modeling community to everyone else, anything we actually do is going to be wrong. We're not going to be able to create a model that resolves all the underlying biology. We're going to miss some physics due to those types of approximations. What we're hoping to do is create a model that we can generate some results or some model that can address some very targeted questions. So we know we're going to make some simplifications, but we're going into this knowing that so we can address where those simplifications actually lie. So where the story starts for Tomopteris is we can glean some kinematic data from movies that are out there, or in our case, reaching out to some collaborators. And you can start doing some digitizing of tracking of motions, measuring morphology, and things like that. So this data, we really want to come from the animals themselves. So we can create sort of a simplified computational geometry where we pull what we think are the interesting or salient features of the movements as well as morphology to address certain things. So in this case, we're really interested in sort of the interplay between the paddling as well as its bodily undulation. So some of the simplifications you may see from our model on the right-hand side is we've gotten rid of the antennae altogether. We've also gotten rid of the more complex head morphology, as well as if you are a fan of Tomopterids, there's about 45 different species or so. Some have very long, elegant tails. We're not addressing the tails whatsoever in our model. So we're really looking at a very specific aspect of parapodia paddling as well as the bodily undulation. And this computational geometry now, it looks like a movie, but if we just stop the movie and look at one frame, and you zoom in, you'll notice that this geometry is actually just composed of 3,000 or so individual vertex points. So this whole model is nothing but a collection of points. To give you a glimpse of what's going on under the hood, at any moment in time, if we want to forward from one frame to the next frame, we need to map all those points, all 3,000 of them, into their next kind of appropriate configuration. So the only way we're able to do that is use as much kinematic data that we can get from these actual organisms. So digitizing all of these movements, the morphology is really important for our line of work, just so we can make sure that, hey, we know what type of movements we're doing, they're realistic, or we know where we're making those simplifications against where the animal what the animal actually does. So if we do this enough times, in our case, we have about 50,000 frames or so that we curate for one complete swimming cycle. What I mean by swimming cycle is one full, basically, power stroke and recovery stroke of parapodia that's timed with its one undulatory cycle of its body wave. And just like if we have 50,000 frames, you can flip through them like a flip book. Or what I want us to start thinking about this geometry is thinking of it more like a wind-up toy. So for example, we have this wind-up toy of this little scuba diver. This scuba diver toy is just doing this leg kicking motion, okay? Any forward or backward, up or down swimming that happens with this scuba diver toy is solely due to the swimmer going like this. We're not telling it how fast to move forward, how fast to move backwards. It's just legs doing all the work. So in that very same spirit, our computational model is just going to try and go through the same geometric states over and over and over. And any forward swimming or backward swimming that happens in our simulations is fully emergent based on the movements of our Tomopteris and the water that it's being immersed in. And the way in which we're doing this on the mathy side of things is using what's called a penalty force framework. So the idea being, if our Tomopteris is not in the correct geometrical configuration at a specific moment in time, there's a little force just to pop it into place. So when we actually do this in our model, we think about creating a geometric representation that sort of averages all of the morphology and kinematic data that we have. So we sort of have what I just called the biologically average case on the top left. But if we want to start playing this game of varying all these morphological traits and kinematic traits, we want this process to automatically, we want our code basically to automatically pop out all these different geometries and cases that we need. So if we want to start changing things like parapodia length, we basically want a little knob in our code just to change that length, change the wave number, change the number of parapodia in general. So we want this process to be pretty automated. So there's a lot of fun optimization going on under the hood. Another mathematical way to talk about this is we need our models to be highly parameterized, where just one parameter tells us things like changing body weight, changing wavelengths, changing parapodia number, things like that. Right? So we sort of have this overview of how we can construct the geometry. And full disclosure, this is just one way to kind of do these types of studies, is through this kind of frame-by-frame penalty force framework. But we have yet to talk about the actual underlying water that we want this thing to swim in. So this comes into play of how we actually couple the motion of this geometry to something like a fluid that we think about it swimming in. So this is where our computational fluid dynamics model comes into play. For us today, you can see what's about to come. Don't be scared. We're going to be using the Peskin's immersed boundary method. And before I get into some of these equations, I want to give you a nice little historical overview of this method. So this method was actually created in the early 1970s to study blood flow past heart valves. Due to technological limitations of the time, the simulations were under-resolved and you couldn't get too much biomedically relevant data from these types of things. However, we're in 2023, and immersed boundary is still one of the leading methods to do this type of fluid structure interaction types of studies. So it's a really robust method that's been improved upon over the years, and it's been used for numerous applications outside, of course, cardiac dynamics. We're doing biolocomotion. You can do some aerodynamic studies, anything in which you have an object sort of interacting with a fluid. Okay. But what the method actually does, it tries to couple the equations for an incompressible viscous fluid with an object that's moving inside of it in some way. So if we start looking at all the characters on this screen, we have the top two equations here, which are your Navier-Stokes fluid equations. They're a nonlinear coupled set of PDEs, or partial differential equations, that describe basically the fluid's velocity at different spatial locations at different moments in time, as well as the background fluid pressure. On the mathematical side of things, there's actually a nice bounty out for these equations. If you can prove some nice properties about their solutions, it's $1 million. So if anyone's feeling particularly inspired, have fun with that. But on the physics side of things, these are conservation laws, the conservation momentum, conservation of mass as well. And in fact, if we look at the momentum equation for a bit, this is actually just a restatement of Newton's second law, kind of written in a more continuum framework. So Newton's second law, the mass times acceleration of an object, equals to all the forces acting on that object. So if we have a little fluid blob or fluid parcel, the left-hand side of this equation describes the mass times acceleration, the so-called quote-unquote material derivative of that blob. The right-hand side are all the forces acting on a little blob of fluid. So from things like pressure forces, which are your normal forces or perpendicular forces to a blob, to your viscous forces, which are your friction-like kind of shearing forces, to this other kind of catch-all term that I'm just calling an external force term. This could be something like gravity. In a more geophysical fluid dynamics context, this could be something like a Coriolis force. For us today, this is going to be the force from a swimmer pushing on the water it's trying to swim in. So it's through this last term that we're able to complete an information exchange about forces from our swimmer onto the water. And that's basically where these other two equations come into play, is we want to do some information exchange between swimmer and water, water and swimmer. So if we take a gander at these two equations for a moment, the first thing you might notice is there's this integral sign sitting in there. This is the same integral from calculus. So all we're going to be doing is adding up a bunch of stuff in some way or another. The top equation is basically going to be doing information exchange from our swimmer onto the water. So what we're actually wanting to do is if I'm swimming in a pool, what this top equation does is add all the force contributions from my movements of my arms onto the water that's nearest to my arms. So that's what these other little delta functions inside of these integrals are doing. They're basically playing this game of, if this is my arm that's moving, what fluid pieces are closest to my arms to spread that force out to? And the other equation does sort of the information exchange the opposite way, from water onto our swimmer. So if we are all playing in the beach, maybe not today, and we're playing in the waves, what this equation tries to do is tell you how the wave motion is going to affect you as some object inside of the ocean. So it's going to basically interpolate velocities from the water onto our swimmer. So on the mathy side of things, another thing our group does is we create open source software for the general community to do these types of studies. So one of our software packages called IB2D is an immersed boundary in 2 dimensions. It's written in both MATLAB and Python in 2 independent versions with about 75 built-in examples to kind of illustrate some of the frameworks and infrastructure in the code, from kind of your prototypical fluid dynamics examples to different types of biomechanics and mechanics applications as well. But all this to say at this point, we have our geometry. We have our fluid dynamics method. The next thing is to put them together. So from the mathematical modeling standpoint, there's still some decisions to be made. So for example, if this is our Tomopteris in the left of this black tank, we have a choice of how big a tank we actually want this thing to swim in. How tall, how long do we want this tank? Another thing we get to choose is the underlying grid resolution. And when I'm talking about grid resolution, you can think of this just like a camera's image. Basically, if an image of just like a PNG, JPEG, if it has a too low of resolution, it's probably going to be blurry. You're not going to be able to see things very distinctly. Whereas a really high resolution image, you can see a lot of very intricate details, but you pay the price of more storage and other fancier type of methods like that. So what we're really looking for is that ever so famous Goldilocks zone in science, where we want to run simulations that aren't under-resolved, because we want to capture the underlying physics. We also don't want to over-resolve because we're wasting resources in both storage and time. So we want to run simulations that are resolved enough that we can capture the details we're interested in. So all that to say, on the mathy side, we do a lot of due diligence tests in terms of making sure our tank or computational domain is big enough, as well as resolved enough for these types of studies. But there's one last thing that I haven't talked about yet in terms of this model. Ben? Validation. That's totally on the spot. So we still need to validate this against what we actually see in biology or can observe with this animal. So this is just one glimpse into one way people do this. Also, full disclosure, this method is not just for validating our type of studies as mathematicians. You should study these animals actually using this method as well. So you can bring both of these types of problems together to kind of study something more holistically. So one way to kind of do this validation or just study the behavior of organisms with fluid dynamics is using a method called particle image velocimetry. So full disclosure again, this is a very sophisticated method that deserves its own lecture series. So I apologize, I'm not going to do this justice in about 3 minutes. But the main idea for this method is you have a tank with your organism in it, the purple piece representing our polychaete, and we also want to seed this tank with neutrally buoyant particles. Neutrally buoyant meaning that any type of fluid motion, these particles are just going to go with the flow and not impede the actual fluid direction in any way. And what we want to use is a high-speed camera to pick up the trajectories of those particles as they move around. Now, the only way to actually do this, to illuminate these particles, is to use a high-intensity laser. So we're setting up sort of a hard problem where you need your organism really to cooperate, because you have your frame of view from your high-speed camera, you have your laser sheet that's cutting into the tank, and you need your organism to kind of swim right through that laser sheet at the right angle you want it to pick up the underlying dynamics. So there's a lot of moving parts in these types of experiments, and they're incredibly complex as well. But one video that one of our collaborators gave us, so this is a group from Mbari, is this is Tomopteris swimming left to right in this tank. The red color here is because they used a red laser, and you might be able to see some of the motion of some of the particles in the background fluid. So this is a raw video image. So what we can do with some PIV software, so I used OpenPIV to do some post-processing on this. You can change the contrast so pick up the particles a little better, but you can also mask the actual organism. So as you're trying to track the particles in the software over time, the software knows not to basically track those particles through the organism, so you don't get some artifacts. And the other thing I'll just mention is due to this being a 2D kind of plain light sheet, we lose information at the top, because the organism is basically blocking information from its other side. So we're only going to be looking at the dynamics on one side of this Tomopteris. But from here, using this type of software, you can track these particle motions in a high-speed way to basically see what the underlying fluid is doing. So I'm just going to show you a snapshot of what the fluid vorticity looks like in this case, and more or less what's happening with this organism as it's performing a power stroke. So the big push of the parapodia, it's basically curating a vortex to form right behind it and push off after the power stroke. And as the parapodia all come together during its recovery phase, you can see it's basically just passing the vortices down along the body. Okay? So that's some of the underlying physics that we're seeing inside of this animal. The other types of things I'll just mention right now is, compared to our simulations, each of the parapodia here are doing something slightly different than the parapodia next to it. Maybe that's changing a paddling angle. Maybe that's just a little bit of different timing of some of its power strokes and recovery strokes and things like that. Our model does not address those types of differences. So all that to say, biology is really complicated. Okay? But this is a snapshot of just one simulation where you can see basically the same type of physics being displayed. We're seeing more or less generally the same power stroke curating or creating a vortex, and the recovery stroke process kind of passing them down along the body. But once we have sort of a validated biomechanical model, this is where we can start asking some actual questions that we're interested in. So one question you might just wonder is, well, how does swimming speed vary with just length of its parapodia? So we could run a couple simulations like this. We can also visualize all of the data. In this case, this is background vorticity. So these are little swirls as well. And you can just observe and see what's happening in the underlying system. And now I know I've been going pretty heavy on vorticity in this talk. I just think it's fun to look at. But we can look at other type of fluid dynamic data, from magnitude of velocity to the actual velocity field itself, the pressure. We can even do things like Lagrangian coherent structures and stuff like that as well. But you can actually see sort of what's happening in the fluid in a qualitative sense. So you can ask maybe some more insightful questions and dig into some more of the physics. But if I'm being honest, we rarely do this more than 1% of the time. A lot of what we really do with these types of simulations is really dig into data analysis, whether that's through time series or whether that's through some type of spatially averaged or time averaged data. So if we think about average swimming speed and we vary a bunch of different simulations of parapodia length, you can sort of see what the effect of varying length happens in terms of affecting its, in this case, swimming speed metric. Now this looks like a terrible plot. It's all to make this point. We varied one trait of the swimmer. We held everything else in the system constant. I didn't tell you anything else about what this swimmer's doing. In this case, it was using a very modest kind of active paddling amplitude. But now at this case, we can start varying that paddling amplitude as well. So in very much an experimental way, kind of doing systematic variations of traits. So we can start varying 2 traits at a time, changing the paddling angle, and seeing sort of what those net effects happen in unison with changing parapodia length. And if we do that enough times, we can fill out a whole 2-dimensional subspace of data, where the colors here represent different swimming speeds, and each kind of box is a unique combination of a parapodia length and a paddling angle. Now, in this case, you might notice there's a lot of boxes here. It doesn't look like we needed that many boxes to actually see what's happening in this system. All that to say, we don't know until we actually do these types of experimental simulations, or experiments or simulations. This is swimming speed, but some other metrics we may be interested in, cost of transport, other efficiency metrics, may show more degrees of nonlinearity. And we want to make sure we're running enough simulations that we don't miss interesting interactions of traits. But there's some challenges to this. Not only did we run too many simulations for at least this type of performance metric, each of these simulations takes about 2.5 days to run on a computer. It also requires about 2 to 3 gigabytes of data if we want to store a lot of data for post-processing. So all that to say, we can't be running simulations all willy-nilly. We have to kind of approach these problems in a more kind of a thoughtful way. But the biggest caveat here is we've only varied 2 traits of this swimming system. If we think about this swimming polychaete, there's many interesting morphological traits or kinematic traits we may be interested in. And in fact, a lot of the kinematic traits sort of give us a glimpse into more of the behavior of these animals. And what I mean by that is these aren't just interesting modeling questions or theoretical biology questions we're interested in. When we actually go out and start observing what these animals do, there's just lots of variation in terms of morphology as well as how these animals are behaving, from things like just size of the animal to even things like what the average paddling angle these things are doing. So with our simulations, we want to try and address this type of diversity. And not only that, there's, like I said, 45 or so species of Tomopterids. This data is only from about 5 species that were collected off of Monterey Bay, hundreds of meters below the surface. So if we really want to start thinking about Tomopteris as a general species in a greater context, we want to include a lot of this natural variation to see what the effects of this type of variation is on performance. And we can even go into a greater context like that because there's other types of holopelagic polychaetes that are fully in the water column their whole life. And you can see Tomopteris in the upper left kind of have very long parapodia, but something like the Alciopids right next to it have kind of very short, stumbly parapodia. But all of these animals are highly maneuverable and highly successful for what they do. So there's interesting biomechanics questions you can start asking in terms of this context as well. So if we start thinking about how we have approached simulations before, this is kind of the design of experiments. I think we run into the same factorial problem that experimentalists do as well. So if each box here represents a unique combination of traits, and we want to start varying 1 trait, well, you can just line up the boxes. If we do this for 2 traits, we saw this type of thing before. Combinations of traits up and down, vertically and horizontally give you unique combinations. And all this to say, if we think, oh, we're going to go in, maybe we vary 10 traits in each dimension. This is already 100 simulations. If we go into a 3rd dimension, we're looking at 1,000 simulations, 10 cubed. If we go into 4 dimensions, you can stack all these cubes just sideways, 5 dimensions vertically. And now that 2D subspace that we saw before is just one slice through this entire huge parameter space. So as you can see, there may be some really interesting interactions of traits, whether kinematic, whether morphological in nature, that lead to other types of very informative performance metrics that also lead into more of a context of the underlying ecology. So the punchline here is these simulations, just like experiments, they take a lot of time, but our punchline in this case is we've been able to do this as a group for varying 8 traits holistically. So there's a lot going on in this slide. All this to say, if you gave me a unique combination of those 8 things I've highlighted, right now our group can say, what the performance should be like for Tomopteris without needing to run additional simulations. So we've basically been able to curate this entire parameter space in 8 dimensions for certain performance metrics. And that's where our group has leaned into in the past couple of years, using kind of surrogate model frameworks that fall under the umbrella of some machine learning. So again, to kind of give you a glimpse of where we're heading, where we're coming from to where we're going, the previous way we've done this in a very much design of experiments way is you think of the traits that you want to start varying. What things are actually going to be your variables in your study? For us, if we're trying to investigate things in very high dimensions, many, many traits, we just can't run that many simulations. That same factorial problem. So full disclosure, a lot of computational fluid dynamics studies have only looked at combinations of 2, maybe 3 traits due to this type of limitation. But using things like surrogate models allow us to build basically an underlying function that will represent the response surface of how performance gets affected by variations in those traits. So in other words, a less mathy way maybe of saying it is we have a function that takes 8 inputs, all the traits we're interested in, and it's going to map those inputs into some performance metric, swimming speed, efficiency, acceleration that we're interested in. So we're really building some type of mathematical function. And before I start casting this all in the language of some machine learning stuff, I want to try and give you maybe a nice parallel to something you may be a little more familiar with. So if we take a quick side view into linear regression, finding the best fit line through data. So if we're given some data in the form of X and Y here, you might want to fit a best fit line. The reason I'm going through this, in terms of machine learning language, we have a model function. That model function being that line that we want to find. Inside of that model function, there are 2 unknowns, 2 coefficients we need to find, the slope and the intercept, M and B. The idea being, you give me different M and Bs, I give you different lines. But there's one line that so-called is the best one. Now, in a mathematical sense, when we talk about best, underlying that hood is some optimization procedure. In the case for linear regression, what we're trying to do is minimize the sum squared of the residuals. So in other words, there's some function that tells us what the discrepancy is between our predictive model, in this case, the line, and the data that we're trying to fit it through. So in a machine learning language, this sum squared of errors is what we call a loss function or a cost function in some way. So when our group approaches this type of surrogate modeling framework, we try to basically fit the data exactly because our simulations, they're completely deterministic in nature. If I run a simulation today, the results I get will be the same as if I ran that simulation on Saturday. There's no stochastic elements in our simulation. So our group has done this surrogate modeling with things like neural nets of various architectures, to other things like polynomial chaos expansions, radial basis functions, Gaussian processes. But underlying all of these machine learning models is some mathematical function that has some unknowns in it. So in this case, all the terms I highlighted are just like the slope and intercept of those lines. Those different values I give you for those, if I gave you different values for those things, we would have a different model fit in some way. So just very quickly going over some of this stuff, maybe it hopefully brings it closer to home. There's basically 2 different flavors of these types of optimization problems when trying to find these unknown coefficients. A so-called linear and nonlinear problem. If we look at the polynomial chaos first, the reason I'm showing this is we have these unknown coefficients, those alpha j's on the right side. We find these actually through the same exact thing that happens with linear regression. This is a least squares problem. We use something like the pseudo-inverse singular value decomposition, call it a day. So pretty standard methods for finding this stuff from some of these models. But something like a neural net is a little more complicated because those unknowns all depend on each other in a nonlinear way. They could be multiplied together and things like that. So because of those types of nonlinearities, we approach this with a little more obscure type of method. Not obscure, but a little more complex. In this case, if you've heard of the language of backpropagation in neural nets, things like gradient descent or stochastic gradient descent, all those methods are basically just trying to figure out what your best coefficients are in the model. In very much the same spirit as linear regression. Right? As well as for neural nets, the other terms I've highlighted in blue here are other things you as the user get to decide. So there's a little more kind of control in that way as well. So all this to say, in terms of our Tomopteris model, looking into the actual study we did, for our training data set, we did about 5,000 simulations sampling that 8-dimensional space. For our test data set, we curated independent data of 1,500 other simulations using a different type of sampling strategy, and we made sure there were no points that overlapped with each other. And you can kind of see that the computational investment was pretty large. So we're not running this on one computer, we're running this on a supercomputer on many nodes. So here's some of the punchline here, because one important thing for any type of these statistical frameworks or machine learning models, you want to make sure your predictive model's able to predict on new data it's never seen before. So just to orient you on this plot, this is a plot of different model evaluations, different simulations, where we're looking at one performance metric. This is average speed. So this is what we call the real data from our simulations. In a moment, I'm going to show you some highlighted points. And these are what the neural net predicts. So hopefully you agree with me that at least in the eyeball norm qualitative sense, there's a lot of pretty good overlap in this case. But of course, we do some other quantitative statistics to make sure our model's actually working the way we think it is, as well as assess where's the modeling error coming in from the neural net. The biggest punchline here is, well, 98% of the time, our model's accurate to under 5% relative errors. But the other important thing is we can actually assess where those larger errors are creeping in. In this case, if you're wondering, for at least for average speed, they're creeping into average speeds that are very close to 0. So overall, some faster polychaetes in these types of worlds are like, I'll just say 0.3 or so body lengths per cycle. This is where the model is basically predicting something like 0.005. And then it's really against a 0.004. So we're seeing large relative errors in that way. Or we're seeing relative errors creeping into the edges of our overall parameter space, just because those edges are less represented in the training data set. So we actually do have some time to get into some results here. To give us a point of view here, we have an 8-dimensional space due to our surrogate models that we can explore any way that we want. So there's kind of a open book on how to actually do this. So there's a lot of data mining techniques that you could use. For one way, you could do sort of the bottom-up approach, where you can kind of explore combinations of traits very much in the original way that you might go out naively and do. Because we have this surrogate model, we can just quickly predict combinations of traits and what their performance is. So you can do this kind of from that ground-up approach. The other approach is doing it from the top down, basically looking at entire performance landscapes where every combination of trait is simulated, and you can kind of see where different combinations of traits overall lead you. So there's a lot of different ways you can approach this. I don't unfortunately think I have time to go through all of this. If anyone wants to discuss stuff with Beer One later, I'm happy to. But I want to close out the story that I started with. The story of why is this Tomopteris wiggling its body if it seems to be pulling it downwards and paddling seems to be pushing it upwards. So you can kind of do this from the bottom-up approach. We can start very quickly combing through different 2D subspaces of parameter combinations and see what performance looks like. So this is a plot of swimming speed versus parapodia length that you saw before. And what I'll just draw your attention to is looking for the longest parapodia between a 50-degree to 60-degree paddling amplitude. You see about a 35% increase in speed. So all that to say, we see a big increase in speed. There's no free lunches in biomechanics, so a system must be compensating in some way. In that way, it's doing more energy expenditure. So one proxy that we can use is defining a mechanical power type output. And if we look at the same data, in this case, power versus parapodia length, each curve's a different paddling angle. Those same 2 points, to get a 35% increase in speed, required over a 70% increase in power. So all this to say, when we're looking at this type of data, we want to better kind of incorporate this data into a larger ecological context, because we're sort of seeing maybe what these polychaetes are able to do, but probably it's not swimming full speed. Just like if we're moving around as humans, we're probably not trying to move as fast as we can all of the time. So there's more behavioral elements that we want to incorporate to these types of studies to ask questions of, why are animals actually doing what they're doing in a more mechanical way? But we can define efficiency type metrics like a cost of transport, which is basically your what are you putting in versus what are you getting out metric. In this case, we're putting in the mechanical power and we're trying to get out something like swimming speed. And if we look at that type of metric, we want to have a minimal cost of transport. And I'll just draw your attention to the white curve here. The white curve is for the largest paddling amplitudes we're looking at. And there's an interesting thing you might notice on this curve. Paddling with the largest amplitude seems to be the best kind of energetic or minimizing cost of transport method here until, we get to a transition region of this around where these parapodial lengths are about 10% or so of the overall body length. Now, why I'm stopping here is because we really only see Tomopterids with parapodial lengths greater than this kind of 10% body length, or this 10% ratio. So really, it's from this 0.1 and over that we're really more seeing Tomopteris. So the question is, so are they just paddling very large and doing something very energetically costly? But we haven't explored how other traits may affect its ability to do things. So if we look at a different subspace now, we sort of hold a very large paddling angle, but now we start investigating the influence of its amplitude and its body wiggling. So this is a cost of transport plot for different parapodial lengths, but for different amplitudes of oscillation. And the interesting story here is this white curve is the largest amplitude of oscillation. So you can kind of see, well, using that high amplitude oscillation doesn't look very good until you reach that transition point. And all of a sudden, after that transition point, it looks like paddling, as well as using a big undulatory wave, really starts minimizing your cost of transport, at least in this parameter space. But this is, it looks like one way that Tomopterids are able to mitigate their cost of transport is if they want to use large paddling angles, it's using a bigger body wave as well. So we can kind of start addressing very targeted biomechanics questions in that way. We can also start seeing how sensitive those results are to other variations in other traits as well. Fortunately, I think I'm running out of time, but I'd be happy to chat afterwards if anyone has any other questions about how we investigate data in these high parameter spaces. But in general, for surrogate modeling, some of the things that you can do with this, once you have a surrogate model that's trained across a big parameter space, you can really start addressing targeted trade-offs in performance, seeing how the system maybe is able to achieve the same performance levels, or see what price they are in some other way, compensating for to achieve those same performance levels. And you can also start addressing questions of how natural variation, particularly if you start sampling your traits from very specific distributions that you see and observe from actual organisms, how those influence the outcome and basically the performance distributions as well. Some of the future directions we're always hoping to do is we always want to collect more and more morphological data, kinematic data for varieties of animals. So all that to say, experiments and experimentalists, we are indebted to them as math modelers to do anything that we do, and doing more of that type of stuff, as well as more gaining more taxonomic data, is also going to help our investigations. The other types of things we're pushing for on a math front is always pushing towards higher dimensional spaces. So we're looking at improving surrogate model frameworks, creating better sampling strategies, and we also do things in terms of creating and modifying our fluid dynamics models as well. So hopefully this talk didn't have too much math in it, but before I lose my math credentials, here's all the math that I swept under the rug for you folks. And I'm really, really indebted to a lot of folks around who have contributed to this work in a variety of different ways, as well as all of my undergraduate students from throughout the years. So thank you very much for inviting me to your seminar. I'd be happy to try and field any questions that you have.
Thank you so much for that. I appreciate you being here. We're going to bounce back and forth. Roseanne, do we have any questions online? Okay, so for folks online, if you have any questions, go ahead and put them in the chat box and we will work through them. Any questions in the room? All right, hang on, I'm coming to you.
Thanks. Great talk. I'll even forgive going to UNC.
Duke?
Duke, yeah.
Gotcha.
That was fascinating. I had a question about investigating these things for, say, a very large vertebrate that you couldn't put in a tank with a laser field, and sort of, I guess, isolate gray whales with drones, and they are in the shallow water and they're doing crazy movements and kinematics that are fascinating, and sort of what approach you would take to sort of trying to study that, even though we could never sort of do the same sort of lab experiments to validate data that you can do with.
So we're talking about manatees, or? I'm just kidding. Whales, right? So whales or sharks in general, the very kind of megafauna. So yeah, it's hard to do those types of studies in any lab setting. I'll kind of give you what I know of the experimental ways that people have approached that. Of course, yeah, like you said, using drones to just capture actual kinematic data of them in the wild. The way that people have approached this in a more laboratory setting is trying to scale down the system and using some type of physical model representation. So there's lots of folks like George Lauder's group, I think, does a lot of this type of stuff at Harvard, but a lot of people have done this for sharks, whales, dolphins, and things like that as well. From the math modeling standpoint, people also do this type of stuff using computational fluids. Some of the, in terms of the model systems that I talked about, in terms of polychaetes, some jellyfish, we're looking for systems that sort of have various symmetries in them. So we're able to do these in 2 dimensions, which helps us do these highly parameterized studies. For something like whales or other very 3D motion type animals in general, it's a little more hard to do these types of big dimensional studies, but people certainly do these studies for thinking about, in a very, very specific way, how does one trait start affecting performance if you have a very biologically accurate morphology. They might just not be able to do a lot of these morphological variations. But yeah, there's a lot of folks that do this type of stuff.
Great, thank you.
Yeah.
Looks like we have a question online, Roseanne.
What is the ecological relevance of this work?
Oh, great question. So I guess I can give you my perspective coming from the math side. So a couple things to address. I'll kind of try and meet in the middle from this answer. So from the math side of things, I think we're always interested in developing models that can say something in nature. And for a long time, at least in what I've seen in fluid dynamics, we're just thinking about things like swimming speed, looking at efficient swimming type metrics like that. However, there's this whole arena where we can start to see, well, how do actual kind of predator-prey interactions take place? How do these animals kind of use their actual kinematics for something else besides just going forward or backward in the ocean? So I think in terms of casting these problems in a larger ecological context, there's some interesting questions that we might be able to think about the physics of how these predator-prey interactions actually come together. In terms of maybe something like the Tomopteris chasing an arrowworm or a small larval fish that it preys upon, how that actual process goes, what it takes to actually maneuver to catch those things, and kind of casting it in that type of problem. And there's we're thinking about doing work like that actually using some agent-based models as well. If you start thinking about the spatial scales where you have a larger animal capturing a smaller animal, we're probably not able to resolve all of those spatial scales in our simulations, but we can start thinking about these smaller animals, particularly maybe some smaller zooplankton, using a more agent-based framework. We can give them a set of rules and we can see how well those rules actually allow them to escape from predation and things like that. So we can look at sort of 2 different lenses of how animals begin to interact.
Nice.
Hopefully that helps a bit.
Okay, 1 second here. We've got a question in the room.
I should also say that agent-based work is with Christopher Strickland at University of Tennessee and Laura Miller at Arizona.
Thank you. This is really cool. I was wondering if, in thinking about like more freshwater entering marine systems, if let's say in those systems the increased freshwater, the decreased salinity didn't kill these animals, or the change in temperature didn't kill these animals, have you looked at modeling changes in sort of the fluid?
Oh, gotcha.
And responses that the animal might need to make evolutionarily to maintain efficiencies?
Yeah, that's a great question. So I haven't looked at really, let me try to address this a couple ways. So please stop me if I'm rambling. But in terms of the underlying fluid dynamics, one of the scaling parameters we use is something called the Reynolds number. I think I have some slides somewhere here. But yeah, so if we have something like the Reynolds number here that can kind of tell us about how the size as well, or a characteristic size, characteristic velocity in our system, as well as meshes with sort of some of the other fluid properties. This is also one of the ways that people can scale down experiments from like a whale into a lab setting. But I'll just say in terms of this Reynolds number, we can do things like changing the density, changing the viscosity of the fluid, and see how variations in that type of scale can affect swimming. I would think an interesting kind of, maybe this is a more behavioral thing for the animal, but would be if we notice if there's any noticeable differences in the way the animal actually swims itself between these different environments. Because maybe those changing kinematics would be, the system might be more sensitive to those changes in kinematics than changes in scale, if they're very small kind of salinity changes and things like that. But yeah, there's a couple different ways to kind of address those types of questions.
All right, looks like we got another question online.
Yeah, great talk. Do the hydrodynamic approach work with smaller organisms like diatoms?
Oh, great question. Yeah, so there's a lot of folks that do this type of thing on even like phytoplankton type stuff even. But yeah, where the actual kind of underlying fluid dynamic story comes in is depending on your size as a critter, you have to develop or you've evolved different ways to actually locomote in your fluid environment. Can I show another movie? Is this okay? All right, so if we're thinking about this type of thing, for example, you can do these types of studies on diatoms, and I'm blanking on the people's name I know that look at these types of fluid dynamics. But you can think about if your animal is in what we call a low Reynolds number type environment. So left-hand side, think about your animal being very, very small. This could be on the order of microns or tens of microns. Now, if this animal is trying to do something like just a caudal fin kind of flap in the back, we basically run into some issues with just how the animal perceives its fluid environment around it. It feels very, very sticky, very, very thick, that this usual kind of fish-like motion, the reciprocation motion, will just kind of undo the process before it. So all that to say, if we take the same exact model and put it in something like water, where we assume through the Reynolds number gives us a lens into viewing that animal as something larger, maybe instead of microns, we're thinking about an animal being on the scale of centimeters here, it just very happily swims across the tank. So all that to say, you can do these types of fluid dynamic studies, but animals have a very interesting lens that they see the world through, and your models can take all this into account.
Nice. Any other questions online, Roseanne?
Yeah, have you looked at the effects of climate change with kinematics?
Oh, that's an excellent question. I personally have not. I would love to get hands on some of the kinematic changes that people have noticed in terms of just how different animals are behaving or what they're doing, but I haven't had my hands on any of that data to start modeling.
All right, I think we're going to call that the end of today. But why don't we give our speaker, Nick, one more round of applause.
Thank you for inviting me again.
And for everybody in the room, I think there was an open invite for some beer. And I think if that is the bartender listening to the conversation that might happen there tonight, that would be really cool. So go ahead and join if you've got additional questions. Enjoy your break, and we'll see you in 2 weeks, everybody. Thanks, everybody.